Good evening and welcome to this lecture on microscopy and staining. I merged microscopy and staining together because before observing the microscope, we usually stain, and whenever we stain, we need to visualize the microscope. So they are partners in the study of microorganisms, so it's difficult to separate the two. So always have to treat them together. Microscopy and staining. So the first section of the lecture will be devoted to describing the different types of microscope and their performances. And the second segment of the lecture will be describing staining techniques, beginning with how to prepare a smear for staining. So we have different types of microscope that we are going to look at. But the first thing I want you to note is that a microscope relies on the principle of contrasting images to be able to visualize an object. We need to we need to produce a contrast between two different images or two different substances that are directed towards the lens to be able to get an image of interest. So basically we have the microscope that will have a lens. We may have a single lens in a simple microscope. We may have two different lenses or more in compound microscopes, and most sophisticated microscope like our electron microscope have more embedded lenses to produce a more magnifying object. So saying that thanks to the contrasting ability of a microscope, thanks to the different lenses, I'm sure you have some basic knowledge of physics, and you know that a lens magnifies an object. So when depending on the magnification of the lens, when the object is magnified, we can be able to see it in a more bigger form thanks to the lens. So when we use more than one lenses at a time, we only increase the magnification and our ability to see the object. The next thing that happens is that because it might be difficult in some cases to produce a contrast between the object and the background, we may have to stain. Okay, so when we stain, we increase the contrast. We increase the contrast and improve our visualization of the object. So we're going to see all of this in the course of this lecture. So the first microscope we want to appreciate is what we call a light microscope. The first category is light microscope. And a light microscope basically produces a bright field under which we are able to visualize the object. So a light microscope produces a bright field, and under this bright field, we can visualize an object. There are two types of light microscopes that we're going to discuss. The first is what I call a simple microscope or a simple light microscope. This one has just a single lens, and it is similar to even a hand magnifying glass. So when you put the object under the lens, you simply have depending on the magnification of the lens, you see the object magnified by that fold. I'm sure some of you might have seen a hand lens. If not, well, go to the laboratory, you have the opportunity to use a manipulate. So a hand lens also magnifies an object depending on the magnification power of the lens. That's the same thing with a simple lens, a simple microscope. But I'm not sure they are still being used now, except in the most remote areas where you find a simple microscope used. They have been outdated because of the sophistication on the procedures we want to carry out. And therefore, we want to go and look but at a compound microscope. Remember, we're still on a light microscope, which produces a bright field. for us to observe our object. So the typical light microscope that we are going to be using in the laboratory during practicals is what we call a compound light microscope. A compound light microscope has a series of lenses that magnify the object, and the final object is a product of the magnification of the different lenses through which the specimen passes through. In some cases, we might immerse in oil to increase on the resolution of the object. And typically of light microscopes, we'll have a condenser that will direct the light onto the object so that the lens can now pick the object and magnify. I have here an image, a typical photograph of a light microscope. As I said, this is almost exactly what we have in the laboratory there that we'll be observing. I'll quickly go through some of the parts so that you'll have an idea of what a light microscope looks like. The first part that we want to note is what we call the ocular lens. If you can see, there are two of them. This is a binocular. In some cases, we might have just one ocular lens. So if you see one or two, it doesn't change anything. This one is a binocular because it has two ocular lenses where you can place your two eyes to visualize image. So remember, I said a compound microscope has different lenses, many lenses that magnify the object. So these ocular lenses are some of the lenses that will improve on the magnification of the object, but this is not where the magnification begins. It actually ends here because this is where your eyepiece is placed. Then now we have the body of the microscope. The body of the microscope is just to handle the ocular lenses and also transmit the image so that we can easily visualize the ocular lens. Then we have the arm. The arm is where we carry. This is where you carry a microscope. You put your hand here and you put the other one under the stage. So this arm here is where you carry the microscope and you put your other hand beneath here. Then the objective lens. This is the first set of lenses where magnification begins. These objective lenses have different magnifications on them. If you look at it, there are 4 of them, or in some cases 3, but I think it's mostly 3. We have times 10, we have times 40, and then you have times 100. So you always have to choose the magnification that you want to use, depending on the object. And when we're doing typical laboratory practice, I always tell you which magnification to choose. But typically, whenever you have to immerse in oil, whenever you're observing under oil immersion, magnification is by default 100. So you have to select the 100 objective. So the first magnification begins here, and then it's again remagnified here, and you observe with your eyes. Then we have the stage. The stage is where we put this is a microscope slide. This slide contains our sample. So it is placed on the stage, adjusted onto the objective lens, from where it is transmitted onto the ocular lens for visualization. Then now I spoke of the condenser. This is what directs and focuses light through the specimen. This is a specimen on top of this glass here. This is light source here, illuminator. The illuminator generates a light source that passes through the diaphragm through the condenser, and this condenser directs this light source through the specimen. From the specimen now, the objective lens picks it up, passes it through the body, and then finally to the ocular lens. That's how the transmission goes. We may also want to look at the diaphragm. The diaphragm is used to control the illumination. If we don't want much light onto the stage or into the specimen, we reduce it using the diaphragm. If we want more light, we can increase it using the diaphragm. And then, of course, the illuminator, our light source. In the typical simple light microscope, this illuminator might have but a mirror that reflects some light and directs it through the condenser to the stage. So this one is uses electric current. So when you plug it on current, this bulb comes on and then directs light through the diaphragm, through the condenser to the stage. Then we have what we call the adjuster. There are 2 adjusting knobs. We have the coarse focusing knob and the fine focusing knob. The first bigger one is what we call the coarse focusing knob. This knob is used to move this stage up towards the objective lens and down away from the objective lens. When you move the stage towards the objective lens, you are trying to pick the image and visualize the field. And at one point, you have to stop, because if you move further, you might instead block the field. So as soon as you start seeing the field, we now use the fine adjusting knob or fine focusing knob to pick the detailed image and then visualize. So this is the typical shape of a microscope. And as I said, we'll go to the laboratory, I'll present this to you, I'll review it, and then I'll let you manipulate it so that we can see images. This is a more 3D image that I think you can better appreciate. This is again still a binocular compound microscope. You see the two ocular lenses where you place your eye. These are the objective knobs or objective lenses where you adjust to pick up which magnification you want to use for the specimen. This is where you place a specimen on this stage. This is our condenser. This is our lamp to produce illumination. That is it with compound microscopy. The next light microscope that we're going to look at is what we call a dark field microscope. Remember, under the first category of light microscope, we had two: bright field microscopy, and I said bright field microscopy is because we have a bright field to which we can visualize the object. Here we have a simple microscope and a compound microscope. The next one now is a dark field microscopy, but remember we are still under what? Light microscope. So this is a dark field microscope, and the difference is that what? The object appears bright against a dark background. The object is illuminated against a dark background. So basically, light rays are refracted as they are impacted onto the object. Those of you who still did some physics, you know what refraction is. The scattering of light as it moves through one medium to another. So as they are refracted now, those that are focused onto the objective lens produce the image, while those that are scattered away from the objective lens produces a dark background. So we are able to see a clear image against a dark background in dark field microscopy. This is an example of it. So we usually use a dark field microscope when we want to visualize very pale objects. When we want to visualize a pale object, we use a dark field microscopy. Before I forget, we use the compound microscope in all staining procedures. So anytime we do normal staining, we have to use but a compound light microscope to visualize. So a dark field microscope, we don't need staining. We just need a pale object we put through, and then a bright object is visualized against a dark field. Next one is what we call a phase contrast microscope. A phase contrast microscope. Remember, I began this lecture by saying that the microscope produces a contrast between the object and the background and can now visualize the object. But there's a specialized microscope which really takes advantage of this ability to produce a contrast between the object and the image. And in this case, we are able to visualize what? A bright image, while the light rays that are scattered away are produced or are focused as a dark background, almost similar procedure to the dark field microscopy. Now, the difference here is that a phase contrast microscope can help us visualize a living object. So a microorganism that is actually motile, we might want to observe motility, how the organism is moving. Use but a phase contrast because we don't need to stain. We don't need to stain the object to visualize, because staining usually kills the organism, and we cannot use it for further analysis. We just visualize and end there. But a phase contrast microscope, we can visualize a living object and move further with our analysis with the object. So we could differentiate two different types. Phase contrast microscopes are indicated, and there's also differential interference contrast microscope. You might never come across the differential interference contrast microscope, but it's very common that you might see a phase contrast microscope. So these are images produced by the three categories of microscopes that are three of the four categories are highlighted. This is a bright field. Here you can see objects, but usually we improve visualization by staining. So these are bacterial cells that you can see here. Most of the time you see them either as purple or pink. So depending on whether they are gram positive or gram negative. We'll be looking at this shortly. Then this is a dark field microscopy. As indicated, the lights are diffracted away, and as they hit the object, so we have a dark background against a bright field. This bright field, these are our objects. These are objects that I wanted to visualize here. These bright substances you see here are our objects, while the background appears dark. This is a normal, typical phase contrast microscope. As indicated, it is used to visualize living objects, while an interference contrast microscope is also used here, but this is more detailed, and as I said, is not very common. The next category of microscopes, we remember the first one was compound microscope. The second one was dark field microscope. The third one was phase contrast microscope. The fourth now is what we call a fluorescence microscope. A fluorescence microscope takes advantage of the ability of some substances to fluoresce when they come across UV light. So when you impact UV light on an object, some of them have the ability to fluoresce, to radiate energy, which is observed as a fluorescence substance. So that is the typical principle of a phase contrast microscope. But in most cases, in most applications of a phase contrast microscope, of a fluorescence microscope, we usually have to stain the objects. So we impact the fluorescence property onto the object so that when we visualize it with a UV light, we are able to observe fluorescence. So let me take it over because of the later confusion. A fluorescent microscope takes advantage of the ability that when a UV light falls on some objects, it produces a fluorescence, and that fluorescence can be observed as a color. But typically in its functioning, we'll always have to stain these objects with a fluorescent dye so that when we observe under a fluorescent microscope, we are able to visualize fluorescence easily. Okay, typical applications are in immunofluorescence, where we used to visualize antibodies produced against pathogens. We can also visualize antigens as well as some proteins. So this is a typical example of an image produced under a fluorescent microscope. You can see that this is an organism. This is a dark background. Now, thanks to the fluorescence ability of the organism, when it encounters UV light, you are able to see a clear color image of the organism. And as I indicated, some organisms naturally produces fluorescence, while with most of the others, we'll have to stain them with a fluorescent dye before we can be able to visualize. These are still images of a fluorescent organism, but these ones are stained and indicated that we could use it to identify antibodies. These are fluorescent. This is a fluorescent dye that has been used to conjugate to an antibody so that we can observe the binding of an antibody to the antigen. So in this case, we have stained this object. So this fluorescent dye is what we'll be able to see after the reaction. But remember that the dye has been conjugated. It wasn't naturally fluorescent. So this image is this is the same image we're seeing here under the microscope. And you can see a very bright coloration across. Now we are done with light microscopes. I'm sure at this level you are able to list all the different type of light microscope and tell me at least the type of objects that will visualize under each of these microscopes. And let me reflect you again. The compound microscope, we want to see objects that we stain. So when we apply stain on this object, we're able to visualize them well. I'm going to talk a little bit more about the typical staining procedures that will apply to visualize object when stained under a compound microscope. Then we had what? A dark field microscope. Dark field microscope is used to visualize pale objects. Then we have a phase contrast microscope. A phase contrast microscope is used to visualize living objects. We want to see them live as they either move around or they behave. Then finally, we had a fluorescence microscope, which I said is used to visualize objects that have the ability to fluoresce. But if these objects cannot fluoresce, we can stain them with a fluorescent dye, like what? Antibodies, antigens, and other proteins. Then now we have electron microscope. Electron microscope here is a more advanced type of microscopy, in the sense that while with the light microscope, which I just indicated, we are doing magnifications of 40 times, 100 times. As a matter of fact, I've been able to see 1,000 times, but all the microscope that you'll be seeing in the laboratory magnifies at most just 100 times. But an electron microscope magnifies an object between 10,000 times to 100,000 times. You can have an idea how big we can now see the object when after this magnification, between 10,000 to 100,000 times. So thanks to this, we can able to appreciate the detailed structure of a virus, a bacteria, internal cell organelles, as well as atoms, even large atoms. There are two types of electron microscopes that are commonly used. There is what we call a transmission electron microscope. A transmission electron microscope is used to visualize the internal structure of cells and organelles. And we also have a scanning electron microscope, which is used to visualize the surface structure of an organism. So scanning electron microscope is used to visualize the surface structure of an organism, while transmission microscope is used to visualize the internal structure of an organism. You can read about the details of this electron microscope. It's beyond the scope of our work. We don't have enough time. So remember I said our lecture was divided into two sections. The first one was microscopy and the second section was staining. Okay, and I said when we stain, here we're really going to visualize with what? A light compound microscope. We are going to visualize with a compound microscope after staining here. So let's focus on staining in this segment. Now, why do we stain? In my very first lecture, I stated that we stain organisms because if I just take a bacterial cell in water and place under a light microscope, it will be difficult for me to differentiate the bacterial cell from water. Okay, the whole background and object will appear the same under the microscope, so I cannot make any appreciation of it. And then second, the fact that bacterial components stain differently. I can also use staining to start differential classification. We spoke last class about gram staining. We also spoke about acid-fast staining, capsular staining, spore staining, and so on. So these different types of staining helps us to begin the initial categorization or classification of organisms. Before we stain, we'll have to prepare what we call in microbiology a bacterial smear. What is a bacterial smear? It's a very, very thin immersion of the bacterial colony that would prepare to enable visualization of single cells on top of a microscope slide. I'll be showing you a smear. I'll be showing you how to prepare a smear shortly. But first, let us appreciate the concept of aseptic techniques, aseptic procedures in a microbiology lab. In our first class again, we admitted that microorganisms were ubiquitous. They were found everywhere and anywhere. So it was typically impossible for you to turn around, swab your table, comb your hair, swab your mouth, your skin, even your foot, and you don't find a microorganism. So because they are everywhere and want to visualize a particular organism that is supposed to be in our specimen, we'll have to always employ sterilization procedures to ensure that we don't contaminate our sample. We don't introduce a new organism into our sample. What we are visualizing is exactly what was inside our sample. Of course, if you have gone through the outline, you realize that sterilization techniques are part of this course model, but that is a lecture for another day. Let me give you a brief idea of what it is. So typically when we culture bacteria, we always have them grow in tubes like this. They can either grow in tubes or they can grow on petri dishes. So on these tubes, they are in a liquid medium. But in another case, we'll have them on a solid agar medium. Of course, we'll see all of this in the next class. I think the next class will be cultivating bacteria. We'll go through typically and see how we can culture on agar medium. But note that we can either have them on a solid surface or on a liquid surface. So let's take the first case where they are on a liquid surface. So when you pick them, the first thing you need to do is you have to check the tube. Why? Because in this tube, there is a disparity of oxygen concentration. Oxygen concentration is highest at the top and least at the bottom. So we are going to have what? Facultative anaerobes growing mostly, but more strict anaerobes might be at the bottom, while facultative anaerobes will be at the top. Remember, the tube is corked, so we are not going to have what? Aerobes. We're not going to have strict aerobes in this tube. Of course, if your target was strict aerobes, you have to open the tube before you inoculate. Sorry, before you incubate. So this is our tube. We have to shake it up. So when we shake it up, we mix up the whole story, and then we pick what we call a wire loop. You also have the opportunity to see and manipulate this. This is a wire loop. This is like our spoon in microbiology, where we dish the organisms to put on the slide. So this wire loop by default is supposed to be what? Contaminated. So anytime we want to use a wire loop, we have to flame over a Bunsen burner flame. We have to pass it over a Bunsen flame to red hot. If you look at it here, you see that it is red hot. So when it is red hot, you allow it to cool a bit. You move it out of the tube to cool a bit. As it's cooling, you are also passing the mouth of your tube toward the flame. Why? Because as it has stood for some time like this, there's a possibility of contamination to have been on it. Or so some sort of contamination to have been on it. So it's ideal that you flame the mouth of the container. So remember, this tube is cooling a little bit away from the flame, but still very close to the flame. Why? Because just by the virtue that there's a heating atmosphere surrounding this flame, it is assumed by microbiologists to be a little sterile. This environment, immediate environment of the flame is assumed to be sterile. So if you cool the loop just around here, there's a lesser possibility that it can get contaminated again. So when it cools and you flame this, you do what? You insert the loop into the tube, and then you pick it out. When you put and remove, when you insert and you remove it out, we are assuming that you have picked about 0.1 microliter of sample. Sorry, milliliters. You have picked about 0.1 milliliters of sample. So when you remove, you flame again, and then look at it here. You use your hand, the hand that is carrying the loop. You use it to cork. You have to cork first before you go on to manipulate what you've picked, because this is more precious to us than this. If this one falls off, no problem, we'll pick another one. But if this one falls off, I bet you when you go to the microbiology lab, you will realize that it might have cost you about 100,000 to get this into this tube. So you cannot afford to let this get contaminated or to let it pour off. So you need to always cork it first. When you cork it first, now you can now come back to this content here. With this content here, this is a microscope slide. It's really a glass transparent slide. So on this microscope slide, this is what you picked. Remember, from the loop, from the tube, you place on the middle of the slide, create a circle, and then you immerse on top of the circle to produce a very thin layer on top of the slide. This very thin layer is what we call a smear. Immediately you finish producing the thin layer, you flame back. Remember, these organisms here could be very pathogenic. So you don't want this loop to contaminate the immediate environment or your immediate surrounding. So immediately you produce a thin layer, you remove this and you flame back. You can keep it somewhere. You now come back to your slide. So I'm assuming that you flame it, you now have back your slide. So from your slide now, in some cases, we advise that you allow to air dry. Okay, you allow your slide to air dry. After it air dries, you can now, in some cases, heat fix. So you pass the slide, the bottom part of the slide or the under part of the slide on top of this Bunsen flame. You could pass it 2 or 3 times. When you pass it 2 or 3 times, in microbiology, you refer to it as heat fixed. Okay, we have heat fixed the slide. So these are YouTube links that you can easily visualize it. I'm going to share these presentations with you, so you take your time. You could watch these. They are just 2 minutes videos. They're very short to watch it, since our laboratory might not be optimum enough to show. I don't have time to be playing the videos now, but their links have been included, so you can play at your time and visualize exactly what is happening. So when you pass over the flame, you have heat fixed. You can now go on with staining. Remember, I was talking about our medium being in a liquid container. On another situation, we'll have our medium in a solid plate. And that is typically what will always be the status quo. Our media will always be from a petri dish. So what do? The first thing we need to do is, again, we do this. We flame. When we flame, we'll pick our petri dish. We're not picking but a tube. We'll pick our petri dish. When we pick our petri dish, the first thing we do is we drop our loop inside water. Usually it's very sterile water that we are working with just beside us, very sterile. We drop our loop inside water and pick a loop full of the water and drop on the plate, on the slide, and we create a circle of the water. Now we flame the loop again. After flaming, we now take the loop into a petri dish. On that petri dish now, we'll pick a single well-identified colony. On the petri dish, we'll pick a single well-identified colony, bring and emulsify into this water drop that we have placed here. When we emulsify, again, we create a very thin layer, just as we did here. We create a very thin layer. After creating that thin layer now, we air dry. After air dry, we'll come forward to fix. After air dry, we can come further to heat fix. We can come further to heat fix, and when we heat fix now, we can keep for staining. Now, why do we heat fix? Why do we heat fix our slides? The main reason we heat fix our slide is to ensure that during staining, our spaceman is not washed off the slide. During staining, our spaceman is not washed off the slide because there's always a likelihood that the repeated washing steps that are part of the staining procedure would wash off our spaceman and we'll have nothing to visualize. So proper heat fixing anchors the spaceman very well onto the slide and it can't be washed off during staining. So after preparing our slides or our smears, the next thing we want to do in microbiology lab is to visualize. Is to stain. So there are different types of staining with different objectives, just as we have with microscopes. We have different types of staining with different objectives. The first one is what we call a simple stain. A simple stain could be a basic stain or an acidic stain. There is a positive stain or a negative stain. And what is the main use of a simple stain? We want to visualize the size of the object. We want to visualize the shape of the object. We want to visualize what? The arrangement of the object. Remember, in my first class, I said bacteria could be arranged in what? Chains, bunches, or in pairs. Okay. I also spoke about their shapes, that they could be spiral, they could be cocci, they could be rod-shaped, they could be what? Comma-shaped. So we can appreciate these shapes if we stain them with a simple stain. We could go further to stain bacteria with a differential stain. A differential state takes advantage of the fact that there's a differential composition in the cell wall components of the bacteria. The most prominent differential stain that we have in microbiology is what we call the Gram stain. The Gram stain broadly divides bacteria into either gram-positive, which have a thick peptidoglycan layer, or gram-negatives, which have a thin peptidoglycan layer. We can also use staining to dictate the presence of a mycolic acid layer in what we call acid-fast staining. The next categorization could be to identify structural components of the cell. For instance, the presence of a capsule, the presence of a flagella, the presence of spores on the bacterial cell. So there are different staining procedures that will help us identify these components. So I said before that stains could be positive or negative. A basic stain is a positive stain, while an acidic stain is a negative stain. A basic stain is a positive stain, an acidic stain is a negative stain. When you look at the typical structure of a cell membrane, what do you have? You have phospholipids tail embedded against what? A typical carbohydrate layer. So what I'm saying is that the typical cell structure of a cell has what? Negative charges on the surface and positive charges buried inside. So if we stain a bacterial cell with a positive stain, we are going to get what? A positive stain that is going to react with the negative charges on the cell. A positive stain will react with negative charges on the cell. And what is going to be the result? Absorption. So the dye is going to be absorbed. So when we stain a bacterial cell with crystal violet, safranin, methylene blue, these stains are going to interact in their positively charged with a negatively charged cell membrane of the bacterial cell and penetrate the cell. On the other hand, if we stain with acidic stain, with negative stains, there's going to be repulsion between the stain and the bacterial cell wall, cell membrane. And because of this repulsion, we are going to get what? A clear layer demarcating the regions of the cell membrane. So you can see clearly the boundaries of the cell membrane. So take note about these two characteristics because we are going to explore them shortly. Let's start with a simple stain. Remember, most simple stains, as I said, are what? Basic. They are positively charged. So when we use them to stain the bacteria, they are going to penetrate the bacteria, and we can observe an object that has absorbed the stain entirely. So we can observe the entire structure of the object. So how do we perform a positive stain or a simple stain? We prepare our smear. I've just described how we prepare a smear in the previous slides. We either prepare it from a broth or from a solid medium. When we prepare our smear, we flush the smear. Remember, we heat fixed. That is the end stage of the preparation of the smear. After heat fixing, we flush the smear with a positive stain. There are so many of them. I identify, say, crystal violet, safranin, methylene blue. We flush the slide with the basic stain. After flushing, we wash it off with water. In some cases, we wash it off with another slide, a cover slide, but typically we have to wash it off with water, wash the excess stain that has accumulated on the slide, on the smear with water. After that, we blot. In most cases, in the laboratory, we don't blot with the blotting paper. We just simply allow it to air dry. Go to a very calm corner, allow it there to air dry. Then after that, we can observe with oil immersion. Remember, I said earlier that with oil immersion, we are observing at times 100 magnification. The next one is differential staining, beginning with gram staining. With gram staining, I've already indicated that want to differentiate the thickness. This is a peptidoglycan layer in gram-positive bacteria, and this is a peptidoglycan layer. You see this thin layer here. This is a peptidoglycan layer in gram-negative bacteria. So, want to exploit to find out how thick is the cell wall layer of the bacteria by observing for this peptidoglycan layer using uh, a gram reaction. So, how do we gram stain a bacteria? I'm taking these two procedures in detail because we have to perform both of them in a laboratory. So how do we gram stain a bacteria? We first of all prepare our smear. Okay, remember it ended where we heat fixed. After heat fixing, we bring the smear. We flush the smear just as we did with primary with a simple stain. We flush the smear with crystal violet. Crystal violet here is our primary stain. Allow it to stand for about a minute. After that, we wash the excess stain out of the smear. Then we cover it with what we call iodine, Lugos iodine. Lugos iodine is a mordant. It helps fasten the stain inside the cell. So after flushing with iodine for about a minute, again we wash the excess. Then we decolorize with 95% ethanol or acetone. This decolorization procedure is aimed at removing this primary stain that we covered the slide with. Is aimed at clearing of crystal violet from the cell. So after decolorization, we come now and we counter stain with safranin for again 1 minute, and then we wash. Now, before I go to the more details, I'll just indicate here that if this primary stain was able to penetrate the cell enough, if crystal violet was able to penetrate the cell enough, the colorization procedure will not be able to remove the stain. And therefore, when we apply the secondary counter stain safranin, it will not penetrate again because a stain has already penetrated. And therefore, the organism will still remain purple. But that is the case of gram-positives with a thick layer. But if this peptidoglycan layer was thin enough, the alcohol will be able to penetrate it, decolorize it, and it will be able to pick the secondary stain safranin, and therefore the organism will appear what? Pink. So this is what I'm trying to explain. At the beginning, we had a presumptively gram-positive cell and a presumptively gram-negative cell. We stain them first with crystal violet, a primary stain. We decolorized with alcohol after passing Lugos iodine over them. Remember that because this layer is very thin, because this layer is very thick in gram-positive, alcohol is unable to decolorize it, and therefore it retains the primary stain, while this very thin in gram-negatives is going to lose the primary stain. And therefore, when we apply the secondary stain, the counter stain, excuse me, when we apply the counter stain, it is going to pick up the counter stain, while the first gram-positives that refuse to let loose of their primary stain are going to maintain that color of the primary stain. So when we observe under the microscope, you are going to see that this is what? A purple bacterial cell. So this is a gram-positive bacterial cell against what? A pink bacterial cell. This is a gram-negative bacterial cell. So I want you, the most important thing is for you to see and identify. This is purple. This is purple. This is a gram-positive cell. This is pink. This is pink. This is pink. This is a gram-negative cell. And I'm expecting that at this level, you know why gram-positives are purple. And you also know why gram-negatives are pink. The next staining procedure is what we call acid-fast bacilli, acid-fast staining, also called Ziehl-Neelsen. Ziehl-Neelsen is a scientist. Ziehl and Neelsen are the two scientists that came together with this staining. aimed at identifying the presence of a mycolic acid layer on microbacteria. So acid-fast staining is typically used to confirm the presence of mycobacteria or Mycobacterium tuberculosis, Mycobacterium leprae. Okay, so I'm not going to describe the procedure detailedly. Take time and feel free to read it. But in the hospital, they always refer to it as AFB. So when the doctor requests for an AFB, know that they are suspecting tuberculosis and they want to confirm the presence of an acid-fast bacilli in your sputum. So this is the way an acid-fast bacilli will appear under the microscope. As I said, you can feel free to read the procedure. I'll just talk also about a negative staining. Also, all this while we talk about the positive stain or the basic stain. Now let's talk about the acid stain. Remember, an acid stain is what? Negatively charged, and it will be repelled by the cell membranes, and therefore we're going to have a clear layer around our organism. Acid stains include Indian ink, necrosine, and so on. Those are all acid stains, and they are particularly exported in confirming the presence of a capsule on bacteria. Remember, in the last class, I said capsules were what? Phagocytic agents. They are agents, organelles that enhance virulence of the organism by avoiding opsonization. So, in a typical procedure, you have to first of all stain the organism with what? A basic stain, a positive stain, so that basic stain penetrates the organism. You identify the organism, and then you now counter stain the organism with what? Sorry, you first of all stain the organism with a negative stain. The negative stain is repelled, identify the typical structure of the organism. We then counter stain with what? A positive stain like crystal violet, sofranine, methylene blue, which will now be absorbed in the organism. And when we observe under the typical light microscope, you are going to see the internal organelles of the cell, the internal structure of the cell stained with the positive stain. Crystal violet should be purple, and then you see this clear layer. This is a layer of the repelled negative stain. So you can now see that there's a capsule, this whitish area. You can now see the presence of a capsule around the organism. This is a more clearer image. So you can see clearly the repulsive area, the presence of a capsule and the organism. This is what you see on the microscope. So this is a slide that summarizes the staining, the objectives of the staining, and what you can see. But note that I've not discussed endospore staining. I've not also discussed flagellar cell staining. Feel free to read more about this, but they are beyond the scope of our lecture. So at this level, I'm going to stop sharing my screen and stop the recording, and we can get on with introduction.